Oh my goodness. And we are back on the ground. Now I'm supposed to be on a 10-minute break. After 2 hours, basically I have 2 hours. I had 2 hours and 10 minutes of break. I just want to take a nap. I'm tired, you guys. I'm tired, but let's keep digging. After this 9 minutes, we're gonna just go back into the studying session, just finishing the reproduction problem set, and then we're gonna get into embryogenesis. Oh God, I don't even know what to say. Though. Hopefully, it passes very fast because I genuinely just don't wanna. Okay, embryogenesis is only like 22 minutes, so in less than an hour, we're supposed to be doing the chapter um and the problem set. Yeah, and then we have the break. Then we have we have 4 hours left. So that's one, that's two, that's three, that's four. Okay. Well, we do have a lot to see. We'll see how it goes. We'll see how it goes. Let's see how it goes. Because otherwise, I would not be studying at all. Still have 8 minutes left. Oh my goodness, I don't even want to do this anymore. We did so far 2 hours out of the six. It's already what 5 p.m. 5:14. Hopefully we get there. I'm done. I'm not having it. I'm having a hard time focusing. I'm having a hard time just staying awake. Not gonna lie, I've been sleeping pretty late for no reason, just scrolling on my phone because I'm just so distracted. Hi. So this is a study with me. I still have like 7 minutes before I start. I'm doing the Pomodoro method, 50 in, 50 minutes in, and 10 minutes out. Right now I'm fully studying biology. So yeah, should get a Jerry curl. What is a Jerry curl? Let me Google that. Jerry curl. Oh yeah, I don't mind my hairstyle. I usually do a twist out. I washed my hair yesterday. But since today I'm not going out, I'm not doing anything. It's supposed to be my rest day. I decided to just keep my twist on. So sorry if it looks a bit crazy right now. Hello, how are you? I'm doing good, and you? I'm okay. Honestly, I'm trying to push through. I have a big exam coming soon, and honestly, it's not the most enjoying situation to be in right now. Jerry girl, girl, be for real. I'm saying it now. I have curly hair. I don't need that. Or like it's a specific type of pattern. Girl, that's so random. That's so random. But I appreciate that. I appreciate the request. Oh, I'm from Gabon. Gabon. It's a country in Central West Africa, just South Cameroon. It's a French colony, so the common language is French, but not the native language. Native language are basically divided into tribes. Um, I mean, I'm not gonna get deep into it, but yeah, we speak French. So yeah. Hello from Hollywood. Okay, Hollywood. I'm from Brazil. Period. Period. Love that. Love to see that you were, watching me from all over the place. That's so nice, honestly. How is Brazil? I've been wanting to visit so bad, so bad. And yeah, it's just that the pocket is not really ready right now. My wallet is yelling at me, so I gotta back down, you know. But for sure, whenever I get the occasion, I'm gonna visit Brazil. I don't speak Portuguese though, but I really want to learn. Hopefully, it's not very complicated. Even if it is, I really want to learn some new languages like Portuguese, Russian. I want to learn Russian. German. I want to learn German too. These are the three languages that I really want to get to learn for sure. Cause French is good. Don't get me wrong. Like my French, it's my native language. English is nice. It's it's very nice. It's the go-to language, you know, whenever you have a language barrier with somebody. Like we usually switch in English, but I really want to learn way more languages for sure. So welcome you guys. If you're new here, it's just a little study session. I was doing a live 2 hours ago, doing the Pomodoro method. Basically, it's just me continuing it right now. But yeah, usually whenever the timer starts, I'm not really looking at the chat or anything like that. So if I don't interact, I'm so sorry. I'm also locking in. Me doing the live right now, it's me not touching my phone. I generally don't want to touch my phone. Whenever I get a break, I usually just tend to reach out for it. And this is how my break cut go from 10 minutes to like 20, 30, 1 hour. So yeah. What is there in America? America is a continent. It's a lot. The US, you mean? I'm not from the United States. I'm just an international student. I'm here under visa to learn. And yeah, that's all. You're back. Hey girl. Yes, I'm back. I'm back. I'm dragging myself here. I genuinely didn't want to be here. Like genuinely. I'm forcing myself to study you guys. Like I'm forcing. You are from oh yes, I'm not from here. Oh, we're just getting started. I've been here for like almost 10 minutes. Hasn't been that long. I have 2 minutes left before we start the study session. We're in biology. I was finishing the problem set about reproduction and problems. Yeah, the reproduction problem set. Same girl, I'm studying bio right now as well. It is dragging me. It is! No, honestly, I don't even know what to do. It's tiring. You want me to teach you how to speak Portuguese? That is Brazilian. That is Brazilian? But for sure, I definitely want to learn. I want to learn Portuguese. It sounds good. It sounds very good. Hi. I'm trying to study. I'm trying to study. We're doing the Pomodoro method. 1 more minute before we start. Hi. So the thing is that now, you might hear like the video, going, speaking and whatever. So if you want to study with me, you're more welcome to do so. I would say if the sound is bothering you, you can still like mute the video if that works for you. But yeah, it's a lot. So much information. But that's because I was dragging it and not being consistent. So now I'm trying to catch up in the last minute, like last minute right now. You say, yeah, thank you. I'm trying my best. I'm trying my best. Trying to show up even when I don't want to. I actually don't want to. 20 more seconds. Good luck on the next session, babe. Um, how much more session you have left? That's a good question. I don't even want to know. I'm scared. I'm just scrolling and more video keep popping up. Um, dang, I don't know. And there we go. We gotta start. I have a bunch of section. I might have to come back tomorrow and do it again. We'll see how it goes. All right, you guys. I'm gonna be focusing here. But if you have any questions, anything, you can leave it into the comment section. And yeah, whenever I get the break, I'll be able to come back to it and respond to it. Okay. Yes, I'm praying to get an A plus for sure. Praying to get it. Okay, there we go. Reproduction.
Cells are identical to each other. At the end of meiosis 1, the daughter cells are identical to the parent cell. This is also incorrect. While it's true that daughter cells from mitosis are genetically identical to the parent cell, meiosis 1 produces daughter cells that are not identical to the parent due to crossing over and independent assortment. Also, daughter cells at the end of meiosis 1 are still diploid with duplicated chromosomes. So C, incorrect. D says during metaphase of mitosis, centromeres are present directly on the metaphase plate. During metaphase of meiosis 1, there are no centromeres on the metaphase plate. This is the correct answer. In metaphase of mitosis, centromeres of each duplicated chromosome are aligned directly on the metaphase plate. In metaphase 1 of meiosis, the homologous chromosomes are paired up and their centromeres are on opposite sides of the metaphase plate. They're not aligned directly on it. And this reflects that fundamental difference in how chromosomes align and segregate. So the correct answer for problem 11 is D. Problem 12 says, which of the following is true regarding prophase? A says the chromosomes separate and move to opposite poles of the cell. This is incorrect. Chromosomes separating and moving to opposite poles happens during anaphase, not prophase. B says the spindle apparatus disappears. This is also incorrect. The spindle apparatus forms in prophase. It does not disappear. C says the chromosomes uncoil. Also incorrect. Chromosomes coil and condense in prophase. They do not uncoil. Uncoiling happens later during telophase. D says the nucleoli disappear. This is true. This happens during prophase as the cell prepares for division. This is part of breaking down the nucleus to allow chromosomes to move. So 12 is D. Problem 13 says, an individual who is phenotypically female is found to have only one copy of a disease carrying recessive allele on the X chromosome, yet demonstrates all of the classic symptoms of the disease. Geneticists determined that the individual.
No,
I'm in high school and I had absolutely no idea what I want to do for my career. And that's what I would have said if I never used FutureScape. It's a personality quiz that actually matches you to.
That the individual has a genotype that likely arose from non-disjunction in one parent. What is the likely genotype of this individual? Okay, let's think about this. We're told that the person is phenotypically female and they show symptoms of an X-linked recessive disease. It only has one copy of the X chromosome. Usually females are XX and would need two copies of the recessive allele to show symptoms. So if she shows symptoms with only one copy, she must be missing the second X, meaning no second normal copy to mask the disease. This points to Turner syndrome, where the genotype is 45X. Only one X chromosome due to non-disjunction. This is an error in chromosome separation. So the correct answer for problem 13 is C. Problem 14 says, during which phase of the menstrual cycle does progesterone concentration peak? Progesterone is secreted by the corpus luteum after ovulation to maintain the uterine lining. its concentration peaks during the luteal phase, which follows ovulation. If no pregnancy occurs, then progesterone levels drop, and that triggers menstruation. So the correct answer here is going to be C. And with that, we can move into our last and final problem. This one says, which of the following would not be seen during pregnancy? Let's go ahead and work through each option. A says high levels of HCG in the first trimester. That's true. In early pregnancy, HCG levels rise sharply so that they can maintain the corpus luteum, which is necessary to keep progesterone production going until the placenta takes over. So this one is normal during pregnancy. B says high levels of progesterone throughout the pregnancy. This is also true. Progesterone is crucial for maintaining the uterine lining and preventing contractions, so it remains elevated the entire time. Choice C says low levels of FSH in the first trimester. That's also
Wait, what is FSH again? Hormones secreted by the hypothalamus to do what? For ovarian follicles.
FSH is the hormone that stimulates follicle development, but once pregnancy begins, you don't want new follicles developing, so it's going to be suppressed. Then choice D says high levels of GnRH throughout the pregnancy. This one is incorrect. This stimulates the release of luteinizing hormone and follicle stimulating hormone from the anterior pituitary, but during pregnancy, both of those are suppressed. So that means GnRH is also suppressed due to negative feedback from high estrogen and progesterone. So the correct answer here is D. This is the hormone that would not be high during pregnancy. And with that, we've completed this practice problem.
Amen. Now we gotta do chapter 3, embryogenesis and what? Okay. Another ad.
Safe like repair, safe like replace.
Wow. Embryogenesis and development.
Hello, everyone.
Why is it a problem set? Oh, okay, she inverted it.
Hello, everyone. My name is Iman. Welcome back to my YouTube channel. Today, we're going to cover Chapter 3 for our MCAT Biology playlist. This chapter is titled Embryogenesis and Development, and here we're going to cover the following objectives. First, we're going to start by looking at the early developmental stages. That means we're going to go step by step through fertilization, cleavage, blastulation, gastrulation, and neurulation. Each of these processes marks a major milestone in the formation of the embryo. We're also going to take a moment to highlight what can go wrong during these early stages and how those issues can affect development. From there, we're going to move into the mechanisms that guide development. So we're going to talk about how cells become specialized, how they communicate with each other, and how they're guided to their proper locations in the body. We're also going to cover what happens when cells are programmed to die, how some tissues can regenerate, and what changes occur as the body begins to age. After that, we're going to take a close look at fetal circulation. This section focuses on how the fetus gets oxygen and nutrients before it's born, and how its circulatory system is uniquely adapted to bypass the lungs and liver during development. Finally, we're going to walk through gestation and birth. We'll talk about what's happening during each trimester, and we'll end this lecture by breaking down the process of labor and delivery. So with this introduction in mind, let's get started with objective 1, early developmental stages. In this section, we're going to explore development from the moment a diploid zygote forms all the way through neurulation, which is the process that gives rise to the neural tube. This is the structure that will eventually become the central nervous system. Let's begin with fertilization. Fertilization occurs at the ampulla of the fallopian tube. This is the widest part of the tube. This is where a secondary oocyte encounters a sperm cell. If you remember from our previous chapter, a secondary oocyte has been ovulated, but it hasn't completed meiosis yet. For fertilization to happen, the sperm has to make its way through two protective layers that surround the egg: the corona radiata, which is the outer layer of follicular cells, and the zona pellucida. This is the glycoprotein rich layer that's just outside of the egg's plasma membrane. This sperm then uses enzymes released from the acrosomal apparatus to break down these layers and make it from the or let's begin with fertilization. Fertilization occurs at the ampulla of the fallopian tube. This is the widest part of the tube. This is where a secondary oocyte encounters a sperm cell. If you remember from our previous chapter, a secondary oocyte has been ovulated, but it hasn't completed meiosis yet. For fertilization to happen, the sperm has to make its way through protective, which is zona happen, the sperm has to make its way through two protective layers that surround the egg: the corona radiata, which is the outer layer of follicular cells, and the zona pellucida. This is the glycoprotein rich layer that's just outside of the egg's plasma membrane. The sperm then the corona radiata
The
which is the outer layer of follicular cells, and the zona pellucida. This is the glycoprotein rich cells, and the zona pellucida. This is the glycoprotein rich layer that's just outside of the egg's plasma membrane. The sperm then uses enzymes released from the acrosomal apparatus to break down these layers and make its way to the egg's surface. Once the sperm successfully penetrates the egg's plasma membrane, its pronucleus is going to enter the egg, and only then does the secondary oocyte complete meiosis too and form a mature ovum and a second polar body. At this point, the egg undergoes a cortical reaction, which is critical to ensure that fertilization only happens once. So, in this reaction, the egg releases calcium ions, which causes the membrane to depolarize. This depolarization has two major effects. First, it prevents additional sperm from fertilizing the egg. This is a process known as polyspermy prevention. The second major effect is that it increases the metabolic rate of the newly formed zygote to prepare it for the next stages of
How did she call it?
development. The calcium release and the membrane changes also lead to the formation of what's called the fertilization membrane. This is an impermeable barrier that makes sure fertilization membrane. This is an impermeable barrier that makes sure no more sperm can enter. Now that we've talked about fertilization, let's take a moment to discuss twins. Twins can form in one of two ways. Either from one fertilized egg or from two separate ones. When two separate eggs are fertilized by two different sperm, we get dizygotic or fraternal twins. These twins are no more genetically similar than any other siblings, and each one is going to have its own placenta, chorion, and amnion. The placenta, by the way, is the organ that facilitates nutrient, gas, and waste exchange between the mother and the fetus. The chorion is the outermost fetal membrane that contributes to forming the placenta, and the amnion is the thin membrane that surrounds the embryo and it fills with fluid to help cushion and protect it during development. When one zygote splits into two embryos, then we get monozygotic or identical twins. These twins share the same exact genetic material because they come from a single fertilized egg. For monozygotic twins, the timing of the split really determines how many structures the twins share. And this has a lot of important clinical implications. Now, if the split happens pretty early,
Alright.
beyond, meaning between 1 to 3 days after fertilization, the embryos are going to develop completely separately. That means each twin ends up with its own placenta, chorion, and amnion. This is called a dichorionic diamniotic pregnancy. If this
Okay, that's where it come from. And twins. to present this. One pasta. I need to repeat that one.
Material because they come from a shared. Then we bring the placenta, and the amnion is the thin membrane that surrounds the embryo, and it fills with fluid to help cushion and protect it organ that or genetically we get dizygotic or fraternal twins. These twins are no more genetically similar than any other siblings, and each one is going to have its own placenta, chorion, and amnion. The placenta, by the way, is the organ that facilitates nutrient, gas, and waste exchange between the mother and the fetus. The chorion is the outermost fetal membrane that contributes to forming the placenta, and the amnion is the thin membrane that surrounds the embryo, and it fills with fluid to help cushion and protect it during development. When zygote splits into two embryos, then we get monozygotic or identical twins. These twins share the same exact genetic material because they come from a single fertilized egg. For monozygotic twins, the timing of the split really determines how many structures the twins share. And this has a lot of important clinical implications. Now, if the split happens pretty early on, meaning between 1 to 3 days after fertilization, the embryos are going to develop completely separately. That means each twin ends up with its own placenta, chorion, and amnion. This is called a dichorionic diamniotic pregnancy. If the split happens a little bit later, meaning between 4 to 8 days after fertilization, the twins may share the same placenta and chorion, but they're still going to have their own amniotic sacs. This is known as monochorionic diamniotic pregnancy. If this split happens pretty late, meaning between 8 to 13 days after fertilization, the twins are going to share everything. There's only one placenta, one chorion, and one amniotic sac. This is called a monochorionic monoamniotic pregnancy, and it comes with higher risks. The split is incomplete. That means that the zygote doesn't divide fully, and we actually get conjoined twins, where the two embryos remain physically connected. And as a general rule, the more structures that are shared, the higher the risk for complications during pregnancy. That's why understanding the timing of the zygotic split is so important in prenatal care. Now, after fertilization, the newly formed zygote begins its journey through the fallopian tube towards the uterus, where it will eventually implant. But before it even gets there, something really important starts happening, and that is cleavage. Cleavage is the name for the rapid fallopian tube towards the uterus, where it will eventually implant. But before it even gets there, something really important starts happening, and that is cleavage. Cleavage is the name for the rapid mitotic cell divisions the zygote undergoes during this early stage. And here's a key point: even though the zygote is dividing, it's not actually growing in size yet. Each division just splits the cytoplasm of the original zygote into smaller and smaller cells. The very first cleavage is especially significant because it marks the transition from a zygote to an embryo. That first division is what officially starts embryonic development. Now, depending on how cells divide, cleavage can be classified in two ways: indeterminate or determinate.
Depending on what?
Embryonic development. Now, depending on how the cells divide, cleavage can be classified in two ways: indeterminate, determinate.
Mm.
Permanent cleavage means that each of the resulting cells still has the potential to become any cell in the organism. In other words, they're not locked into a specific goal yet. This is the kind of cleavage that allows for the formation of identical twins because each cell is still capable of developing into a full organism. On the other hand, determinate cleavage means that the fate of each cell is already set. These cells are committed to becoming a specific part of the body, and they're no longer flexible in terms of what type of tissue they'll form. As cleavage continues, the embryo goes from 2 cells to 4 cells to 8 cells, and so on, all while it's traveling through the fallopian tube towards the uterus. Once cleavage is complete, the next stage in the uterus. Once cleavage is complete, the next stage in early development is blastulation. After several rounds of cell division, the embryo becomes a solid ball of cells, and we call this structure the morula. At this point, the cells are still tightly packed together, and there's no visible internal cavity just yet. But very soon, fluid is going to begin to fill the inside of this ball of cells, and it will form a hollow structure called the blastula. The fluid-filled cavity inside is called the blastocoele. In mammal, fluid, and it will form a hollow structure called the blastula. The fluid-filled cavity inside is called the blastocoele. In mammals, the blastula is more known specifically as a blastocyst. And this is where we start to see some specialization in the cells. The blastula is more known specifically as a blastocyst. And this is where we start to see some specialization in the cells. The blastocyst is made up of two main parts. The outer layer of cells is called the trophoblast. These cells will go on to form structures that support the embryo, like the chorion and eventually the placenta. Inside the blastocyst, there's a group of cells called the inner cell mass. This cluster is what will eventually become the organism itself. Every tissue and organ in the developing body will eventually come from these cells. So at this point, the embryo has gone from a single cell to a structured multi-layered sphere with both supportive and embryonic components. Now that the blastocyst has formed, the next step is implantation. The blastocyst continues traveling down. Fallopian tube until it reaches the uterus. And once it's there, it's going to attach to the endometrium, which is the inner lining of the uterus. This is when the embryo is going to settle in and begin forming deeper connections with maternal tissue. Now, the outer layer of the blastocyst, which we call the trophoblast, these play a key role here. These trophoblast cells are going to start to invade the endometrium, and they're going to form structures called chorionic villi. These finger-like outer layer of the blastocyst, which we call a trophoblast, these play a key role here. These trophoblast cells are going to start to invade the endometrium, and they're going to form structures called chorionic villi. These finger-like projections are going to help anchor the embryo, and they later become part of the placenta. The placenta is what connects the developing embryo to the uterine wall. And through this connection, the embryo receives oxygen and nutrients, and it's able to have waste products removed. The exchange happens through a structure called the umbilical cord, which links the embryo to the placenta. But early in development, the placenta isn't fully functional yet. So before it's ready to take over, the embryo relies on four structures for support. First is the yolk sac, which helps with early blood cell development. Then there's the antubis. This is involved in early fluid exchange. Next is the amnion. This is a membrane that surrounds the embryo, and it fills with fluid to cushion and protect it. Finally, the chorion surrounds everything, and it contributes to forming the placenta. So together, these structures keep the embryo alive and stable during the earliest stages of pregnancy until the placenta is fully developed, and then it can take over. Once implantation is complete and the blastocyst has settled into the uterine wall, the next major step in development is gastrulation. Gastrulation is the process that establishes the three primary germ layers of the embryo. It begins when certain cells on the surface of the blastula start to move inward. This movement is called invagination, and it transforms the blastula. It begins when certain cells on the surface of the blastula start to move inward. This movement is called invagination, and it transforms the blastula into a new structure called the gastrula. As cells migrate into a new structure called the gastrula. As cells migrate inward, this movement is called invagination, and it transforms the blastula into a new structure called. gastrule. As cells migrate inward, they form a tube-like structure known as the archenterion, which will eventually develop into the gut. The opening to this structure is called the blastopore. And here we want to make an important distinction. In humans, which are deuterostomes, the blastopore develops into the anus. In contrast, in protostomes, the blastopore becomes the mouth. As this inward folding continues in tall, rich, great inward, they form a tube-like structure known as the archenterion. Which will eventually develop into the gut. The opening to this structure is called the blastopore. And here we want to make an important distinction. In humans, which are deuterostomes, the blastopore develops into the anus. In contrast, in protostomes, the blastopore becomes the mouth. As this inward folding continues, some of the migrating cells also move into what remains of the blastocoele, which is the fluid-filled cavity in the blastula. This movement establishes three distinct layers of cells. These are known as the primary germ layers, and each one gives rise to specific tissues and organs in the body. Let's go ahead and walk through each of these three layers. The first is the ectoderm. The ectoderm is the outermost layer. This gives rise to the skin, hair, and nails, but it also forms the nervous system, the lens of the eye, the inner ear, and the lining of the mouth. Any structure that interferes with the external environment or has a neural origin typically comes from the ectoderm. Next we have is the mesoderm. The mesoderm is the middle layer, and this one is responsible for forming the musculoskeletal system, including muscles and bones, as well as the circulatory system, the excretory system, and the gonads. Now, the adrenal cortex, which is part of the adrenal gland, also develops from the mesoderm. Finally, we have the endoderm, which is the innermost layer. The endoderm forms the epithelial linings of the digestive and respiratory layer. The lens skin through as the primary germ layers, and each one gives rise to specific tissues and organs.
Great.
in the body. Let's go ahead and walk through each of these, which is the fluid filled cavity in the blastula. This movement establishes three distinct layers of cells. These are known as the primary germ layers, and each one gives rise. To specific tissues and organs in the body. Let's go ahead and walk through each of these three layers. The first is the ectoderm. The ectoderm is the outermost layer. This gives rise to the skin, hair, and nails, but it also forms the nervous system, the lens of the eye, the inner ear, and the lining of the mouth. Any structure that interferes with the external environment or has a neural origin typically comes from the ectoderm. Next we have is the mesoderm. The mesoderm is the middle layer, and this one is responsible for forming the musculoskeletal system, including muscles and bones, as well as the circulatory system, the respiratory system, and the gonads. Now, the adrenal cortex, which is part of the adrenal gland, also develops from the mesoderm. Finally, we have the endoderm, which is the innermost layer. The endoderm forms the epithelial linings of the digestive and respiratory tracts. It also gives rise to the pancreas, thyroid, bladder, and heart. Now, at this point, even though the basic body layers are set up, the cells themselves still need to specialize into their final forms. And this happens through selective transcription, which means only certain genes are expressed in each cell, depending on what it's meant to become. And
Free me. Free me. Oh my goodness. I'm going to pass out. Pass out. Oh my goodness. Oh, well, I'm here. Okay. Um, let me skin you're brave and amazing. You got this. Thank you. I'm trying. I'm trying. Oh my goodness. I feel like I need to find another method to be a bit more efficient when it comes to, you know, doing it because I believe Professor Iman more precisely she provides the PDF of what she goes through. So I feel like I'm gonna just get it if possible. Yes, she has the notes. I'm gonna just get it, go through it with her during the whole video, adding my notes, do the problems because writing down is cool because I feel like when you write it down, it makes more sense. But it's such a hustle. Like I'm really running out of time right now. Oh, okay, she got everything. It's in the description bar of her videos. Nice. Oh, God bless that woman because honestly, if it is just me, or like giving up in a lot of things right now. Okay, Unreal Genesis. Here I am. So I sent it to myself and I put it into my good note. And yeah, where is it? It's only like 10 chapters, you guys. Like in one day, I'm pretty sure I can go through it. We're trying to be delusional. Hello. You are horizontally challenged. Yes, I am. I am. I don't know what that means, but I guess I am. Offline what? I'm offline? What do you mean by that? Okay. Just no, I guess. Mm, let me do that. Oh my goodness. That was too long to be real. I mean, if I need to use Chrome, let me use Chrome, guys. What's going on here? Or I can just send it to myself because at that point, I'm getting impatient. Not gonna lie. How are you guys doing? We got this. We got this. I don't even know what I'm gonna be eating tonight. I'm already hungry. That's something about me. Whenever I start studying, I I snack a lot. I need food. Don't tell me I'm gonna be struggling here just doing that. Or I can just find the video myself because it's taking its free time. My computer is about to die. Um, like it's old. It's a very old computer. Puta, where is it? Mm, which page am I even working it on? And I just pick whoever. YouTube is now doing too much with the ads. Like it's not even enjoyable to even watch a video or anything like that. Cause there is no way I'm gonna be paying for YouTube. Why is everything having ads right now? Like you guys are struggling to get money or what? So God, I need to get Chrome. No, no, there are way more chapters. I'm so confused right now. Oh, now it is. Duh. Okay, let me cancel it there. Okay, I got it now. What's going on now with the music? And there it is. Chapter 3. Nice. I got everything. She is so smart. Like, this is crazy. I don't know what happened with the music. It's just not working right now. It stopped. I feel like I'm already overworking my computer with just having a few tabs open. So I'm gonna. Computer, don't leave me. Oh, I count on you right now. Where did it add it? It okay. Very good. All the notes are there. All I can do right now is just following along, and yeah, we got this. How long is my break? Only have 1 minute left. Dang it. Okay, you guys, I'll be right back. I'm craving cashew. So I'm gonna grab some cashews. 48 seconds. Give me a break. Mhm. That was a good break. Now we're going back into the study session. 50 minutes in, 10 minutes out. Basically, now we already did 1 hour out of the four, which is considerable, I guess. Hopefully. Now I'm gonna play the video. I have all her notes, so we're gonna just go through them so that I don't have to like write them down, because if I take my sweet time to write them, we're like 6 hours is just generally not enough. The two past hours it was okay, but like now I'm running out of patience. So thank God she posted them, honestly. How was the session? It was okay. Right now we're taking another strategy to approach it. I just got her notes because she posts her notes into the caption. So hopefully it worked pretty well. So I'm gonna just go through the notes with her, and after that I'm gonna have to do some active recall of all of them to make sure that it makes sense in my head. Yeah, hopefully it works. Have you tried the blurting study method? No, I have not. I don't even know what that is actually. Let me write this down so whenever I'm done studying for the day, maybe before going to bed, I'm gonna do my research on it. Cause that's good. I really appreciate when you guys give me some tips like that. You know, studying can be such a hustle. Sometimes I just don't want to be here. I do the whiteboard method. Um, you know, writing everything out onto a big whiteboard. I like to emphasize on big whiteboard because I feel like I have space and it's a mind map. Then I do it's basically me taking notes in the form of a mind map, and after that I do active recall, and the way I do it is like I'm teaching an imaginary class. That's how I do it, and so far it's been sticking with me. But when it comes to setting like that, the Pomodoro method is actually the least, in my opinion, like the one that I don't really like, because I feel like when it is cutting me off like 30 seconds in and whatever seconds out, I mean 30 minutes in and whatever minutes out, I don't know, it's kind of blocking me from the flow. That's why I opted for the 50-10, because I want to have a sort of a full hour in, but at the same time I also need a break, so it helps me getting a break, so I feel like I just need a longer period in. So far, the 50-10 works. So I'd rather be doing that, I guess. But I would definitely try the blurting study method. I'm gonna look that up as soon as I finish. And yeah, thank you so much. Let me see. What was I even looking for? Oh yeah, the video. And I'm gonna write this down too. Okay, I'll check that out. Basically a type of active recall study technique. Oh, that looks good. That looks good. I'm gonna look into it into details more. My remember one is kind of like blurting, but it's after you studied or reviewed the content you write. Oh, or you remember out in your head. True, that's actually great because the way I do the whiteboard method was more like I was taking notes on the board while doing the lecture. So I would not, just watch the lecture and then writing down um the information that just got stuck with me. But that's actually a good method right there because it will actually make your brain work. Mm-hmm. You might be putting me on something, actually. That's a good one.
Derm, which is the innermost layer. The endoderm forms the epithelial linings of the digestive and
Where are we?
respiratory tract. It also gives rise to the pancreas, thyroid, bladder, and parts of the liver. Now, at this point, even though the basic body layers are set up, the cells themselves still need to specialize into their final forms. And this happens through selective transcription, which means only certain genes are expressed in each cell, depending on what it's meant to become. Another key concept here is induction. This is when one group of cells influences the fate of nearby cells through chemical signaling. The cells that receive these signals must be competent, meaning they're capable of responding. The molecules responsible for this signaling are called inducers, and they often guide nearby tissues towards specific developmental paths. Now, once the three germ layers have been established, the next major step in embryonic development is the formation of the nervous system. This process is called neurulation. The first structure to form is the notochord, which develops from mesodermal cells. Now, the notochord doesn't become part of the nervous system itself, but it does play a critical signaling role. The notochord sends out molecular signals to the ectoderm, which is the outermost germ layer, and it causes a specific region of it to thicken. This region is called the neural plate. As the signals continue, the edges of the neural plate are going to move inward and fold inward and essentially form what's known as the neural fold and the central neural root. These folds continue moving towards each other until they meet and fuse and essentially form a sealed hollow structure that's called the neural tube. This tube will eventually give rise to the central nervous system, which includes both the brain and the spinal cord. Now, at the edges of this folding process, we also form a specialized population of cells called the neural crest. These cells break away and migrate to various parts of the embryo where they're going to develop into a wide range of structures. This includes the peripheral nervous system, pigment cells, part of the adrenal medulla, and connective tissue in the head and neck. Now, one final note here, this stage of development is extremely sensitive to outside influences. Certain substances, known as teratogens, they can interfere with these early developmental processes. These include things like alcohol, drugs, certain infections, and even environmental toxins. Exposure to these during early pregnancy can cause some serious birth defects, or in some cases, even lead to the death of the embryo. With that, we've completed objective one, and now we can move into objective two, which is titled Mechanisms of Development. The human body contains around 37 trillion cells, and almost every one of them is specialized to perform a specific function. From neurons to red blood cells to skin cells, every type of tissue plays its own role, and each cell knows what to become thanks to a tightly controlled sequence of events. This process starts with specification. In this stage, a cell is reversibly designated to become a certain type. So it hasn't fully committed yet, it's more like a suggestion. That changes in the next step, which is called determination. Here, the cell becomes irreversibly committed to a specific thing. This decision is guided by chemical signals called morphogens, and the ability of a cell to respond to these signals is known as competence. After a cell has been determined, it begins to differentiate. Differentiation is the process by which a cell changes its physical structure and its internal machinery to carry out its specific function. So, for example, a cell that's destined to become a neuron is going to start building the proteins and the structures that are needed for transmitting nerve signals. Now, before differentiation begins, many of these cells actually start out as stem cells. Stem cells are undifferentiated cells with the potential to develop into a wide variety of other cell types. And depending on their level of flexibility, or what we call their potency, stem cells are classified into three main types. First, we have totipotent stem cells. These are the most flexible. They can become any cell type, including cells from the three germ layers and extra embryonic structures like the placenta. So a fertilized egg and the cells of the very early embryo, they fall into this category. Next are pluripotent cells. These still can form any cell from the ectoderm, mesoderm, or endoderm, but they can't make placental tissue. Finally, we have multipotent cells. These are the most restricted. They can only become a specific subset of cell types, usually within the same germ layer. Now, once cells begin differentiating, they don't just work in isolation. In order for tissues and organs to form correctly, cells need to communicate, and they do this through a variety of chemical signaling methods. The first type is autocrine signaling. In this case, the cell releases a signal that actually acts back on itself. It's like the cell is giving itself instructions, and this kind of feedback can really help reinforce a developmental decision or amplify a response. Next is juxtacrine signaling. This happens between two cells that are directly touching. This includes signaling through gap junctions where molecules pass directly from one cell to another. It's like a shared bridge for quick communication. Then we have paracrine signaling, which occurs between nearby cells. This signaling molecule is going to be released by one cell, and it's going to diffuse over a short distance to affect its neighbor. This is especially important in development because it allows cells in one area to influence the fate of adjacent regions. Finally, there's endocrine signaling, which involves long distance communication. The signaling cell is going to release a hormone or a chemical into the bloodstream, and then it travels to reach a distant target cell somewhere else in the body. This is the most widespread form of signaling, and it plays a key role in coordinating changes across tissues. All of these signaling methods, they play a role in how cells respond to inducers. Inducers are molecules, often proteins, called growth factors that are secreted by one group of cells to influence another. These molecules trigger specific changes, and they usually promote differentiation and cell division in targeted tissues. Now, a common theme in development is reciprocal induction, where two tissues take turns influencing each other's fate. So, for example, one layer might signal the one next to it to start forming a specific structure, and that second layer then sends a signal back to refine the original. Now, this is so cool to think about because it's so similar to how humans get things done in like a group project or a research lab. We can also see the effect of morphogen gradients. Morphogens are signaling molecules that spread out from a source and they form a concentration gradient. Cells that are close to the source receive high levels, while those that are further away receive lower levels. And depending on how much morphogen a cell is exposed to, it can activate different sets of genes and end up becoming different cell types. Of course, once cells have been told what to become, They need to get to the right place, and that brings us to cell migration. During development, cells often originate in one area, but they need to move to another location to do their job. Migration is guided by chemical signals, extracellular matrix proteins, and sometimes physical barriers happen. A great example is neural crest cells. These cells form at the border of the neural tube, but they don't just stay there. Instead, they migrate throughout the embryo and they differentiate into a wide variety of tissues. So when we talk about development, it's not just about the cell knowing what to become. It's also about knowing where to go, when to go, and who to talk to along the way. And that coordination is what makes complex multicellular life possible. And that is truly amazing. Next, I want to talk about what happens when cells die. That means we need to distinguish between apoptosis and necrosis. Apoptosis is a form of programmed cell death. It's highly organized and it's essential for proper development. During apoptosis, the cell shrinks, its DNA condenses, and it breaks apart into smaller fragments called apoptotic bodies. These are then cleared away by immune cells through a process called phagocytosis without causing inflammation. This helps sculpt tissues during development and it eliminates damage or unnecessary cells. On the other hand, necrosis is uncontrolled cell death, and it's usually caused by injury or trauma. In
Mhm.
necrosis, the cell swells, its contents begin to leak out, organelles break down, and inflammation follows. So it's messy, and unlike apoptosis, it often damages surrounding tissues. Now, another interesting thing that's worth discussing is regeneration. Regenerative capacity refers to an organism's ability to regrow parts of its body. Some organisms, like salamanders or starfish, can regenerate entire limbs. That's called complete regeneration, where the tissue is restored exactly as it was before. In humans, however, regeneration is typically incomplete. We can repair damage, but the new tissue may not fully match the original, and this, of course, varies by the tissue type. For example, liver cells can regrow after damage, but a severed limb won't regenerate. Finally, to wrap up this objective, let's talk about aging. As an organism gets older, both its cell and tissues undergo gradual changes. These changes affect everything from DNA stability to protein function to cell signal.
Mhm.
Over time, this leads to a disruption of metabolism. It leads to reduced repair ability, and ultimately, it will lead to the death of the organism. The cellular level, one of the key features of senescence is telomere shortening. Telomeres are protective caps at the ends of chromosomes. And with each round of cell division, the telomeres get shorter. Eventually, they become too short to protect the DNA, and the cell receives a signal to stop dividing. That's when cellular senescence begins. These cells remain alive, but they don't divide, and they contribute to aging and inflammation if they build up in tissues. With that, we've completed the second objective, and we can now move into the third objective, where we're going to talk about fetal circulation. This is the system that allows the developing fetus to receive oxygen and nutrients without using its own lungs or digestive organs. The key to this system is the placenta. The placenta is a specialized organ that allows the exchange of nutrients.
Ooh, wait, I need a slight break. My neck hurt. I don't know. Yep, I feel like I need to take it out. Yeah, I feel like I need to take a nap. This is crazy. You know when you're just looking at the screen and um like below your eye, it's like you can feel it beating a little bit. That's what's happening right now. I feel like I need to close my eyes. I might be pushing a little bit too much for that 4 hours, but we have what, 30 minutes left? Dang it. Ugh. Okay, I feel like it's enough biology for the day. Let me look at um what can I do now? I can download the notes for sure. I can do that and read it on my own because I feel like I'm gonna do the oh I haven't. Thank you. I have not been drinking water. That's a good one. Dang it. What the heck is going on? This is bad. This is bad. I really don't like school like that. I don't. I just want to run away. Not even drinking water. I'm here dehydrated. Okay, nervous system is supposed to be the next one. Um, respiratory. What? Homeostasis. I'm about to start MCAT prep. Oh, girls, I am scared right now. This is what I'm doing. I'm scared for my life. Oh my goodness. No, I'm not scared. It's okay. It's just that that's why it is important to put a value into what you're studying. Because if you're someone like me who was caring pretty much like about her grade more than the knowledge, this is how you get basically caught up. It's bad. You need to put a value into what you're studying. That means all the time you should ask yourself, why are we studying that? Like, what does it have to do with me or with the environment or whatever? Just put a value into it so that it gets into your head. Don't memorize it. Because I'm a big memorizer. This is not a problem that I'm having because I'm required to use what I memorized as a, this is the basically the application. You need to know where it comes from. Okay, that's great. Because on the MCAT, they're not gonna be like, oh, um, what is the definition of this? Heck no. They'll be talking about a case study, and from there you need to infer to understand what happened, this step, this step. I didn't get it like that. I just know A, B, C, D, E, F, G, that's all. But I don't know why is A, why is B, Here I am now, trying to make it make sense, connecting the dots. Oh, this is just bad. But, better late than never, honestly. Better late than never. It should be like my last time doing that, and then I'm gonna just go straight to AMC and doing full length tests, honestly, because um I don't think I'll be able to master all the topics. That's a lot. So hopefully I'll be able to figure out during the test because I was talking to a medical student and she said the like 80% of the answers during the MCAT are in the test. So you just need to be able to infer to because most of the time you have all the questions are true, but which one is the most true? And that's the part where I'm like, I just don't get it. Like false, true, true, I get it. But all of them are true. One is more true than the other one. That's why I get confused. And most of the questions on the MCAT are like that. You should try recapping the content now to kind of give yourself a break, but still solidify the info. You can try and explain what you remember to me now if you want. Oh, that's putting me on the spot. Not gonna lie, embryogenesis was one of the longest plus reproduction. So I don't think I even remember anything. I'm just going chapters after chapters. But that was a good one, honestly. I feel like I'm gonna take another break, eating some food, drinking more water, and I'll sit down in silence reading my notes, and I do the method you showed me. I'm gonna get my board, and I'm gonna be doing that on the whiteboard because oh my goodness, that was a lot. That was a lot. I feel like I would just go back to the video if after reading the chapter I still don't get it because I'm wasting my time. Yeah, harder than the MCAT. Yeah, but we got this though. I feel like the MCAT is just a matter of pattern. Um, yeah, it's about patterns. And yeah, U word. I heard U word. Yes, yes, U word is. I was first using U word. My membership has expired, and I'm not gonna renew that. It's very expensive. Um, and I hated working in that for some reason. They provide very good materials. I would highly recommend that as a first choice because that's everybody go to. But I'm gonna stick with AMC and Blueprint because they're cheaper. That's the thing. It was just too much. It was above and beyond. I feel like I've spent about $1,000 on UWord, and it's hard. It's just so tedious. It hurts your confidence. Like every time I thought I ate it, I'm looking at the result and I'm like, wow, not even on the average, not even close to the average. I was like 23%, while other people, like the average was like 63. And I was like, dang, you know what, you were? I'm going to take a break from you because that was too much. But if you are remembering after the chapters, then um, we're going to do something. Exactly. We got to do something different for sure. Because I feel like I get it, but not to the point where I can, talk about it. I don't. I still need more time to like sit down and make it make sense in my head. That means I need to sit down and review that again. Yeah, for sure, for sure. Yeah, AMC, yes, it is a good one, and it's the closest you have to what you're gonna be doing during the exam. So yeah, AMC, honestly, nothing to say. I just need to understand the patterns and learning to figure it out because I'm used to definition, you know, copy paste type of answers and questions. Now it's like be more precise, think way more. It's just that I'm not used to critical thinking, so it's fine. It's fine. What have you been doing now? Just watching the videos of the game. Yes, that's what I've been doing. And she also has a problem set. Basically videos where it's kind of a review thing, and this is what has been helping me a lot compared to the whole chapters actually. So basically she has a case study and everything and then the answers. And I really like going to these videos because the way she asks the questions look a bit more like MCAT, if that makes sense. It's not definition wise, it's more like more wordy, but definitely very pretty much simpler than the MCAT. Simpler, but I like the way she builds her questions. So yeah, this is what I've been doing the problem set, and then that's all. Trying to refresh my mind because biology's been it's been a minute. I don't remember biology. Biochemia, but biology, not really. Not really. Are you going live again? Yes, I'm definitely thinking about doing it. I'm gonna just make sure to get all the how do we call that? all the notes and we'll see how it goes. Tomorrow I might be doing the other method instead of doing that. Um, Maybe having the Pomodoro in the back going on while I'm doing the whole, you know. Could you repeat who you were watching? I think, oh, Professor Iman. Yeah, Professor Iman. She's amazing. Just amazing. I would highly recommend you to just go on her YouTube channel. She has a playlist for each sections. Just not the behavioral science one. So no sociology and psychology part. She doesn't have it, but the rest she does. I mean, from what I remember, maybe she has one, but yeah, that's that. I think that's all. Hopefully my computer is still alive. I don't know what's going on. Of course, of course. Thank you so much for your help. I'm planning on starting my NCAT prep tomorrow, so I better. Yes! I'll try to be here. I'll try. I need to go to bed on time, honestly. Because lately I just be having no motivation. But honestly, just brutally putting myself into it, I feel like it has helped a lot. So I might be consistent. I better be consistent because I'm running out of time. Like running out of time. But yeah, we're gonna be doing that for sure. I'll see how it goes, and I might as well just tomorrow doing the live and having my board and trying to do the method you recommended, honestly, because I need to wrap it up as soon as possible. Next time I sit down in front of this computer, I should be doing either a full length test or just a section. Have you found any Anki? Oh, so I don't use Anki because it's too complicated. I'm not really good with computers for some reason. But I use it's called Kony over here. So basically they have a library. I mean, obviously you have to pay for it, but it really worth it. I don't remember how much it was, but definitely less than $15. Um, and they have already made flashcards for MCAT, anatomy, or medical terms and everything. That's something that I can keep for in the future. They also have business, math, geography, physics, and the one that I'm using is the MCAT miles down. I'm supposed to do few today. I'm supposed to do 130 today. It's because I've been lacking. But yeah, it's very nice. That's how it is. That one says, this is an example of blank epithelial tissue. If I remember, that's a transitional epithelial tissue. Yes, it is. And sometimes you have the link of the video right here of a video that explains what it is. So I just click on it. And that one leads me to the Khan Academy. Sometimes it can be on YouTube or not, and they talk about it if you don't really get it. But that's nice. Honestly, love it. Um, I need to check. Oh, of course, you should check that out. That's a very fancy one right here. Cogni, Cogni. Yes, I know the content. I know the content. It's me. Oh my goodness. But yeah, they have everything is mixed up. Um, yeah, but you can also get it into small sections like only MCAT physics, only MCAT organic chemistry. But I rather just having everything together at that point because I'm running out of time now. I'm just rushing. Yeah. All right. Till it is 7 p.m. I feel like I'm gonna be ordering some food because that's just pure gluttony right now. I'm not even hungry. I feel like I just need to do something. So I'm gonna get some food. Thank you so much again for being here. I really appreciate that, and I appreciate the recommendation you made. I'm gonna definitely look into that because I definitely struggle to find some efficient way to study. Yeah, that's why I always start something, I don't finish it because I feel like it's not efficient. I start something else, I don't finish it. So yeah, hopefully it'll work. I feel like it'll work out because I kind of already do something similar. I just need to be more efficient when it comes to doing it. Yeah, sounds good. Thank you again for being here. Two, we did 4 hours out of the 4 hours each. 3 30 minutes out of the 6 hours. Have you tried dark chocolate during your study sessions and nuts and fruit as well? Oh, that's a good idea. I don't really like dark chocolate. I don't really like dark chocolate. I mean, I can do, maybe 70% dark or like balance out like that to have more taste. But I definitely need to get more groceries when it comes to snacks because I eat a lot. I snack a lot, especially when my brain is working. I need to eat. And fruit as well. Yes, I finished all my fruits about a couple of days ago. I love my mandarins and apples. I'm gonna get them again next month. And yes, nuts. Yeah, I just got cashews, but they're salted cashews and salt. Too much salt is not really good, so I usually just grab one handful per day. Um yeah, because I was not really drinking enough water, but eating a lot of cashews last week. I got the worst stomach ache ever. Never again. I'm gonna order Big Terry's, a good burger with some fries because I'm hungry. I don't even have some fries actually. I'm gonna just get the burger so I can heat up the fries in the oven. You know, baked fries for a little bit of healthy, while having a whole burger. Yeah, either pizzeries or Hot Doddy. It's just that Hot Doddy can be a little bit pricey, not gonna lie. So, but I'm thinking. I'll see how it goes. Yeah, the fries never misses, never misses. Hmm. Okay, I feel like I'm gonna have to pull a little overnighter just going through the chapters I was doing today because I feel like I definitely need to go back ASAP before it just ends up not making sense at all in my head. Because at the beginning it was making sense, but the reproduction chapter and embryogenesis, oh my goodness, they're just so heavy. I'm like, you don't even make any sense. That's the chapters that made me hate biology in high school. See, and they're like 55 minutes. They're the heavy ones. Oh my goodness. No, I need to go back into it. But yeah, I definitely need to go back into it before tomorrow, possibly, and then we'll see how it goes. All right. Thank you so much for being here. I'm gonna leave you guys and see you guys tomorrow, God willing. I have to be here, showing up for myself because if I don't do that, I'm literally self-sabotaging. And we're not doing that here, okay? We're not doing that here. Bye bye.